That's great. Welcome everybody and good afternoon in Boston. As Liang told that I'm Anil Kumar and today I'm going to talk about two of our method that we developed recently. One is RNA data. We have named SC type, and then later on I'll show you the use of this very tool to predict drug combinations targeting specific anti-cancer subclones in acute myeloid leukemia patient. So many of you might be aware that yes, now it's better. So many of you might be aware that single-cell RNA sequencing has really revolutionized the process of identifying new cells and their role in disease as well as healthy conditions. For example, some of these recent papers, the first one in Nature Genetics, showing that how single cell sequencing can help this process. And the second paper is a little bit older, from 2019, I think, which characterizes the different cells in human liver tissue. This technology is really nice, and many of you, I'm sure, that might be using. However, the process of cell type identification is not quite easy, and it's hectic. Hectic in the sense that first, it requires a lot of programming skills, and many of the experimental researchers may not have these various skills. Second, it requires knowledge of cell type specific marker. And third, it involves multiple steps of analyzing the data. For example, once you have single cell sequencing data, then of course you do normalizing and then use clustering. You can use multiple clustering ways to cluster your cell type. Once the clustering is done, then the usual process, or at least when we started, then the standard was that you identify differentially expressed gene across cluster. For example, you can do a differential expression between the red level cluster versus all other cluster, and you identify these genes, go back and match to some known either database, or if you have been working in the field, then based upon your own knowledge that what could be the appropriate marker for a particular cell type, and then come back and try to annotate these cell types or clusters, which was kind of okay, but the process get complicated or has its limitations because that many of the cells either lack a particular specific marker, or a marker could involve in multiple cell types. For example, the one here shown in green is a marker for the first cell type, and then the another cell type which has been shown in orange, and even a marker for the third cell type which is slightly red. So it could be that what we previously believed as a marker for a specific cell type can show expression in multiple cell type. And similarly, a marker may not show a cluster specificity which we desire, that if it expresses in only one cluster, then we can say that this cluster belongs to this particular cell type. But usually we see these kind of cases where our non-marker is not expressed in only one cluster, but across multiple clusters. Third problem was that if you try to, it's easy when you have one or two marker, but let's assume that a cell type has been reported to have like 2-3 markers. Then there was no idea how to integrate them and say that what cell type it is. And of course, we all know that the number of markers are limited, and we need more markers to better characterize the cell types. So to take into account these limitations, we thought, we started in 2018, that was quite long back, and we started accumulating the database that were available then. For example, PangloDB was there, cell marker was there, and we perform a excessive literature search in PubMed that were not included in cell marker database, or many company, for example, BD Biosciences and other, they also provide their own marker database. So we went through literature and combined and made our SC type database and curated it manually. For example, we try to go back and see that a database, a marker that has been reported in our database has more specificity as compared to like uh, noise in the literature. So based upon that, we were able to identify marker for 17 tissue and 194 human cell type, total almost 4,000 marker. We have also made a database for mouse, as like these two models are quite commonly used to for single cell type identification. So for mouse, we were able to identify 194 cell types and some 4,200 markers. So till then, till now as well, like this is the largest database for cell type specific markers. And then we try to implement an algorithm that basically take into two things into account. So one was that if a gene has low specificity in terms of expression in a cluster, then it penalized it while scoring a cell type. And if it has a high, like cluster specific expression, then you give a higher score for this kind of marker. And secondly, if a gene is a marker for many cell types, then you give a low score for this kind of marker. And if it is specific to a cell type, then you give high score for this kind of marker. So schematically, you can imagine here that those mark like the one at the corner and has been encircled, where we try to give higher weight for these markers because they were more expressed in a specific cluster, and as well as based upon literature in our database, they were reported as a marker for a specific cell type. Example for our algorithm. So assume that there are 6 genes and their marker, they have been reported as a marker for 3 cell types, for example, T1, T2, T3. So gene 1, which is a marker for all 3 cell types or tissue type, and gene 2 is a marker for only like one particular cell type. Gene 3 is a marker for, gene 2 is a marker for only one tissue type, gene 3 is for tissue type 2, gene 4 is tissue type 3, gene 5 is also for tissue type 3, and gene 6 is for two marker, like two tissue, T2 and T3. So based upon that, we calculate an occurrence in our database. So for example, you can see here that gene 1 has been reported as a marker for 3 tissue. So it is also a marker for T1, T2, T3. So the occurrence is 3. Gene 2 is a marker for only one tissue, T1. Gene 3 is a marker for again 1. So similarly, we give a number for each gene based upon their occurrence in our database. And then we scale this occurrence in the range of 0 to 1. So for example, gene 1 is a marker for all 3 tissues, so its specificity score is 0.
Sorry, Anil, can I ask one question here?
Yes.
Can you, since we have plenty of time, right? So I think everyone are, welcome to ask a question. Just raise your hand and then we can unmute you. So I have one question here. So when you actually have, basically have 2 column markers like plus VE and marker minus VE. Can you explain that? Because for gene 4, we have T3 and T1. What's the difference there? Yeah.
So one of the unique approach that we, sorry for not explaining that, like one of the unique, like all the method use just positive markers. So for example, if a gene is expressed in a particular cell type, so they consider it as a marker for that particular specific cell type. But we thought that even some genes, for example, we expect that they shouldn't be expressed in a particular cell type. For example, if you assume in terms of pancreatic cells, so you expect that insulin should be in the beta cell types, But at the same time, you don't expect that glucagon should also be in the beta cells, at least higher expression of beta cell. So what we thought that if we could this kind of information that is already available in the literature as a negative marker for a particular cell type, then maybe we can increase the specificity of these markers and better annotate the cells. So a negative marker means that you don't its expression in tissue 3. So you don't expect the expression of gene 4 in tissue 1. Did I explain it?
Yeah, great. Thank you.
Okay. Yes. So you scale the occurrence in the scale of 0 to 1. So you can assume that since gene 1 is present in all 3 tissues, so its specificity score is 0. Whereas gene 2 is present in only one tissue type or cell type, then its score is 1. Gene 4 is present in like 2. So in between 3, 1, and 2, it's like a score is 0.5. If you scale it, you'll get that. So the next step was that you have this raw expression matrix from your single cell sequencing, and you Z transform it. So Z transformation is basically the expression of the value with the standard deviation variance of 1 and mean to 0. I think all of you are aware of this transformation. So you transform it, and then you multiply the gene specificity scores to this transform matrix, and you get a weighted expression matrix. And in the third step, if your tissue type has negative marker, then you kind of like subtract. So first, for example, if you have like multiple marker for a tissue type, here in case of like C1 and C2 is one tissue type, which you can so you sum them. And how do you sum them? Is just you take a sum, like average of these multiple marker for the same tissue type. So you, in that way, you can have a positive marker set expression. For example, MS1, MS2, MS3 represent the 3 tissue that I was explaining earlier, T1, T2, and T3. And MS1 was like T1, and it has 3 genes, and the gene was gene 1, 2, and 3 as well. Yeah, no, gene 1 for T1, gene 2 for T1, and then gene 4. So gene 4 is a kind of negative marker. So you add gene 1 and gene 2 and take it average, and then subtract the expression of T1 for calculating this MS1. And in that way, you'll have a like sum matrix, which you can see here at the last, that like based upon the higher expression value, these not about to like the cell types were correctly get assigned to these tissue types. Did I explain it correctly? Like, is there any question on this slide? So basically you average it, and then you subtract the negative marker value, and once you'll have a like the final cell type enrichment score, then based upon the highest score, you define the cell type. So based upon this, we check the performance of our model in published data set. And out of these 73 cell types that we ask our model to annotate, 72 were correctly annotated, which gives us an accuracy of almost 99%. One cell type in the brain tissue was not able to annotated because I think the marker were not available in our database. And then we compared it to.
And yes, one more question.
Yeah.
Can I ask like how many genes you actually use? So if you go to the previous slide, how many genes you actually use to calculate the score? Do you use all the genes, or you actually use the top, the most variable genes?
Yeah. So no. We use all genes. We are not selecting anything from the like raw single cell RNA matrix, as many methods do that. They just take the most variable gene. No, we match the gene from our database to the raw expression matrix, and we take the match.
Got it.
So for example, one cell type, if our database has 10 genes, then we'll extract the 10 gene matrix from. It could be that for some cell types, there are more markers in our database. Some cell types, it could be 3 or 4 or 5. So it totally depends upon how many markers are there in SC type database. So we annotated this and then we compared it, then the best performer. So we selected SC sorter because it was reported the most accurate at that time point, and CNA because it was the fastest, and SCCAS because it was the automated. And our SC sorter was also performing well in many of the data set, but our was also the best one, like almost equal. But our real benefit was in terms of time. SC type was the fastest, and giving the fastest annotation possible in out of the all available method. And the reason was that as compared to other method, which they try to use higher computationally costly algorithm like optimization or k-means clustering, our is a simple scoring method where you do a simple multiplication, like you do a Z transformation and then they multiply by our specificity score, and that gave us an edge in terms of computational cost. So it's pretty fast. And here I saw one example of using the negative marker. For example, if you use only SC, if you use SC type and use only positive markers, then you may annotate the one the cream color cluster here as effector T cells, but actually it should be naive T cells. And the reason is that if you go to literature and see that the naive T cells shouldn't express like cell and the effector T cells shouldn't express cell and CCR7 because they are like more mature and they have been migrated to lymph node. So if you use these kind of negative markers such as cell and CCR7 for annotation of T cell, then you can correctly annotate this subtype of a particular cell type such as T cells more accurately. And that's why one of the benefit of SC type is these negative markers, although there are not for many cell types, but wherever they are really informative to correctly annotate cell types. Another feature what cell types has, and it's like well ahead of all other methods, is that it can also help you to identify which are healthy and cancerous cells if you are trying to use it for this purpose. Like if you are trying to annotate the cells from cancerous tissue, then it can be really informative to suggest you that which of these cells are healthy and which of them are cancerous. So how does it do is that it calls SNV and CNV using the raw BAM file that you obtain from scRNA-seq profile. And we only do it for cancer consensus gene, which are, I think, 580 or some number. We get it from the Cosmic database. It has been already like tagged with our tool, and it calls SNV using Strelka2 and sum it. So this SNV score is nothing but the sum of the number of SNV called in a particular cell. Similarly, we call CNV using Copycat, which was published in 2021, and we assign the cells either diploid or aneuploid. And if a cluster has less than 20 CNV score and greater than 50% of the cells are diploid, then we annotate those cells clusters as healthy cluster cells. and others as kind of cancerous. So we tested this approach in our AML patients data that we have in-house here. So this is one annotations we obtained from SC type. And when we call SNV, then we can see that those T cells here have less SNV as compared to the HCC MPP cells, which are supposed to be cancerous in case of acute myeloid leukemia. And you can see here that as you go from these T cell subtypes to more neutrophils and multipotent stem cells, the number of SNV increases. Similarly, we call the ploidy of the cell types, and you can see here that the T cells are diploid, whereas the multipotent cells and ISG plus cells are more of aneuploid. So it kind of indicates that our method is able to identify healthy, and so healthy means non-malignant and malignant cell in cancer tissue. So the tool is available as a web application where you can upload your own data, and just on 2-3 clicks, you can get an annotation. You can choose your parameters. And for other analysis, for example, filtering and normalization, the tool uses scRNA. So we don't have done any modification over there. It's normal scRNA algorithm that run behind it. The paper is available in Nature Communication. It came this year, and this is the GitHub code in case somebody is interested in running, like not on the tool but their own. Then you can also type Ian Vesky and SC type. So all the tools are available freely. And sctype.app is the web link for this tool. Is there any question till this point? Because now I'm going to show an application of this very tool to predict drug combination. So I'll be very happy to answer if there is anything.
Any question from the audience? So you can either raise your hand or just type your question in the Q&A. Nope, I think we can.
Okay.
We can move forward.
Yes, so we also work on prediction of drug combination to treat cancer, and we work on acute myeloid leukemia cancer, and our target is to predict personalized drug combinations. So as drug combination has become kind of a standard to treat especially therapy resistant cancer, so predicting safe and effective drug combinations are very, very kind of useful and can reshape the treatment of these drug resistant patients. So for optimal drug combinations, the expectation are that first it should be effective, and second it should be less toxic. For example, it should only target the cancerous cells, not the healthy cells. Third, it should be synergistic. So synergistic is that by synergy, it means that if you combine two drugs, then you expect an effect which is better than either of the drug alone. So the idea is that if you combine two things, then you should get some extra benefit by having either of the drug alone. And then fourth is that although you can predict but it should show some, like you should be able to test it into primary cells. So otherwise, just based upon computation prediction, it will be very hard to translate to clinic. So these are the features that you expect from a like optimal drug combination. So we wanted to develop a model that take the single cell RNA profile of a single patient. I'm talking here about personalized drug combination prediction of a single patient, and ex vivo drug response. Ex vivo drug responses means is that you take in case of acute myeloid leukemia, bone marrow cells from the patients, you culture into a plate, and in the presence of a drug, and you measure the growth or growth inhibition of these cancer cells due to the drug as compared to a whale that has not been treated with the drug. So in that way, you can have a estimate of how a single drug is effective against cancer cells. So given single cell RNA and single drug response, can you predict safe, effective, and synergistic drug combination? That was the task here. So to do that, we used scType to cluster cells, and like the cells, we only took the cancerous cells here, and the idea was to match the expression of drug target to drug response. Since we have measured drug response here, the single drug response, we expect that if a patient has a higher expression of the drug target, then that drug should show more effect or efficacy as compared to a drug that has lower expression of the target in a patient, cancer cells of the patients. So based upon that, we kind of develop a model where, as I told, that we have drug response, which is measured in terms of selective drug sensitivity score. It's a score that basically it's area under the curve for treatment effect of drug at multiple concentration. So we try to match this sensitivity or efficacy with their target. So we collected target of these drugs based upon drug target common, which is a tool we have developed, which is a database we have developed here in our lab. IUPHAR, that is a publicly available tool that reports about drug target, drug bank, and DGIDB. And we calculated a target expression score, which is just similar to the GSBA score. If you might be aware, like we used a tool which basically sums the expression of different targets in cancer cells from our patients and put it into a XGBoost model to predict the single drug response here. So once this model is trained, then we ask that if we combine the target of two drugs together as an input, then what's the sensitivity score or this DSS score for this combined drug? So basically, first we train the drug where for a single agent, and then later we combine the targets for drug 1 and drug 2 together and predicted the single drug sensitivity score. And based upon that, we use a model that's called synergy model, which we have developed in our lab to estimate synergy score and prioritize the drug combination that which are going to be more effective. So as I told that you can combine genes which are target of drug 1 and drug 2, and we put back into this XGBoost model. And this conformal learning is to remove the low confidence predictions, and you have this combination efficacy score. You put into a synergy model and you can identify the top synergistic drug. And later on, we also selected these combinations based upon their efficacy score. So what is this selective efficacy score? Is that if you expect a drug or drug combination to be less toxic, then you assume that its target or the target of this drug combination should be highly expressed in cancerous cell, but low expressed in healthy cells. So as I told that we use scType to identify the cancerous cells and healthy cells. So basically you can subtract the target cells to the healthy cells to have a kind of a score that will tell you that if it is higher, then the drug is more likely to be active in the cancerous cells and less in the healthy cells. So based upon that, we selected the drug, and then we tested in 3 patients 4 patients, 4 samples from 3 patients. So the one highlighted here, like AMLD1, AMLD2R, and then 2 samples from the patient 3. We tested it in the same cell inhibition assay that was used to measure the single drug response. And we are able to correctly predict synergistic drug combination 53% of the time. Although the number doesn't look marvelous, but one has to remember that if you randomly combine drugs, then the chances of getting synergistic drug combination is just 5%. So it is much higher than the random combination of any drug, 2 drugs together. Later on, we also tested them in flow cytometry, and we observed that our tested combinations showed low toxicity in the healthy cells. For example, the orange here are T cells, and then the blue here are AML, like AML, which is cancerous cells. So the tested combination, for example, venetoclax, bistusertib, was more effective in AML cell and less effective in T cells, which is the healthy cells. And it was true for like all 3 patients. Like, although it varied from patient to patient. For example, in case of AMLD1 and AMLD2, they were more selective, but for this ML3 refractory samples, like the combination were not as selective as in case of patient 2. So this was one example, and we published this in Science Advances in 2021. Sorry, like it's getting repeated. So at this point, I end my talk. I was about to show only the SC type, but then at the end, I thought that I may show some example of the use of SC type. So if there is any questions of the second part of the talk, I'll be happy to answer. And all these tools are like this. scCom was also openly available tool. It is available in GitHub. So if anybody is interested in predicting personalized drug combination using single cell profile, then you can go back and use it. Like the codes are freely available, and scCom is also available as R package.
Nope, I think that we can save the question later.
Okay, then that's all. Like, okay. I wanted to show that much only today.
Okay, so I basically have 2 questions. So I think the first question is that SC type can be used to do the cell type annotation, and also we can use the SC type to call the the genetic variants, right? So I was just wondering, yes. Did you use the BAM files to do the calling?
Yes, yeah, we use the BAM.
Okay, it only takes like around like less than 2 hours to do the annotation and also the kind of the genetic variants calling, right?
Yes, so it was taking longer time, but then we are trying to write some of these, for example, copycat, some of the steps of the copycat into C++ so that it could be faster. So we plan to make it like 5 to 10 minutes for even annotation. So we are working on that.
I see. So what's the input data actually we need for the single cell SC type to run? It seems like we usually just use the expression matrix, right? But here it seems like if we need to do the genetic variance calling, we have to use BAM files. So
yeah, exactly.
Does that mean that we need?
Yes, you have to upload the BAM files there. Okay,
so both basically.
Yeah.
The other question I have is so for the single cell RNA-seq data, do you think that we can use those single cell RNA-seq data to do some more analysis? Like, for example, in cancer, like gene fusion is kind of also very common, right? Do you have any future plan, like say, for example, include this kind of function to like detect gene fusion, that would be also interesting, So right now, I think you can do like cell type annotation. You can identify the genetic variants, and then maybe you can, if you can, think about anything else, something else like to do, like for example, gene fusions. Maybe that's another interesting function we can use, especially for the cancer, single cell RNA-seq data. I'm not sure you have any other plan for the assay type, in the future.
Yeah, that's very good suggestion. Yes, we are thinking to extend it, like first the database itself, because the markers keep accumulating. So at some point in time, we'll update the database. And second, we are trying to develop it in a way that you can identify the origin of these cancer cells. For example, if you are comparing two samples, let's say diagnosis and relapse, then these cancer subclones or a particular clone in a sample, we are trying to develop it in a way that you can go back to the diagnosis sample and see that where it is originating from. So we are developing it in a direction where it can be used as a standard tool to analyze like multi-time samples. So because nowadays like more samples are being collected for diagnosis and relapse and like time point. So that's currently where we are thinking. But of course, there are many exciting analysis which we can use single cell profile for, and it will be useful. Like it will be exciting to implement all of them. But then at some time point, the tools start to get slower if you have like many more.
Yeah, that's right.
So that's one thing we are kind of cautious about, and then don't try to include everything.
Right. Understandable. Any other question from the audience?
Yeah, audience. Not audience, I mean like users. And depending upon like what's the demand later on, we can think of like implementing those functions in the assay type at some time point.
Great. Yeah, we have one question from the audience. Adam, yeah. So I'm actually going to unmute you so you can talk right now. If you can.
Yeah, so I've not fully understood the calculation in assay type. Should MS1 row in positive table be the same as gene 2? Yeah, if it is like I can go back and then so maybe.
Anil, I think we have one question from the.
Yeah. Adam.
Oh,
right?
okay. You saw the question.
Yeah.
Let me read the question. I'm not sure. Maybe everyone can see the question. I have not fully understood the calculation in the cell assay type. Should MS1 row in positive table be the same as the gene 2?
Just a minute. So the Zoom login is just coming above the table, so I'm not able to see the numbers. Yeah, but theoretically it should be that if there are more than one marker for a marker set, then their numbers should be summed and taken an average. But if let's say gene 2 is only one cell, only one marker for a particular cell type, let's say cell type 2, then its value won't change, it will remain the same. So the idea is that if you have more than one marker, then you average the particular cell type, then it will remain as it is. Did I answer Adam's question?
So does MS1 represent T1?
T1, T2, and T3. MS1 represent T1. Yes.
Yeah. So I think the positive marker MS1 should be the sum of gene 1 and gene 2, right? Or the average.
Gene 1 and gene 2, and then gene 1, gene 2, and then minus gene 4. Yes. the sum of gene 1 and gene 2. Yes.
Yeah, but here for the weighted expression matrix, the gene 1 is all 0 and gene 2 is 1.28, 1.30. But in the positive marker set expression, MS1 is 0.90, 0.92. So does it match?
Right. Yeah, so if we take average, then 0.12 systems. I can, so gene 1 for tissue type 1, we have, yeah, so gene 1 and gene 2 MS1, and before the negative marker it should be kind of sum or a square root. Well, I can go back and Christian, are you there? Do you remember like how the formula should be?
I need to unmute him. Maybe you can unmute him also. Do you know how to do that?
Yeah, I can try. Hello to talk. Yeah, I think I can go back and check into the.
I think I have figured out the calculation in the figure legend.
Okay.
It says it's summing them and dividing by the square root of their number.
Yeah. So yeah, I think that's the one. Yeah, not average, but like sum divided by a square root. You are right. Yeah.
Okay, thank you very much.
Yeah.
Great talk.
Great. Any other question? All right, I don't think we have any more questions. Thank you, Anil. Very nice talk. We really appreciate that. Have a nice day.
Thank you.